you, Dr. Shervan, for the introduction and for helping me make this presentation before the schedule. It was supposed to be tomorrow, but I made a request because I've got to leave tomorrow. And the study that I'm going to be presenting is the preliminary data from our lab in this medical institute, which is northwest of New Delhi, about 200 kilometers from New Delhi, in the foothill of Himalayas. And the data basically revolves around the use of lineage negative stem cells derived from the cord blood, which is banked in all the cord banks which are coming up across the world and also in India, as to how it can impact the rescue of memory loss artificially induced by amyloid beta, which is one of the traditional ways that Alzheimer's disease is known to be caused, apart from neurofibrillary tangles and tau phosphorylation and amyloid beta deposits. So we sought to deliver stereotactically the amyloid beta, A beta 142 into the hippocampal regions, intrahippocampi, and tried to cause amnesia, which is memory loss in mouse, and the entire study is in the mouse. So, and then we tried to enrich the stem cells, and this is the figure. This is zooming into the brain, CA1, CA2, C3 regions, and that's the region where we tried to target and trying to deliver the amyloid beta 142 using digital stereotaxis. And then that's when we thought we could contribute in the disease, in the pathogenesis or in the rescue of the memory loss caused due to Alzheimer's disease. Alzheimer's disease has its own growing number of patients which are coming up, not only in the developed countries such as India, US, Australia and Europe, but also in India, and China, which is witnessing an increase in the lifespan and also an increase in the degenerative disorders of the brain and retina, primarily Alzheimer's disease and AMD, which causes central vision loss. So it's known to be caused due to a variety of reasons which I just enumerated, but in addition, and the oxidative stress and mitochondrial dysfunction is also known to be one of the cardinal features of the pathogenesis of Alzheimer's disease, which we sought to create or mimic in this model of mouse by depositing amyloid beta. And the basic inspiration came from the large number of cord banks which are coming up in our country and across the world, especially in the United States. And we have about 7 banks in India promising therapies. And we thought we should at least do something which is more applicable, at least in the context of the commercial business that these cord banks are promising to donors. And it's between $1,000 to $1,500 that a cord bank charges from the donors who bank their cord blood and the tissues with these banks. So we thought, let us look at the potential of lineage negative stem cells derived from these cord blood, as our lab has been focusing on these stem cells, which are basically endothelial precursor cells. We believe they have a lot of CD34 and CD45 positive stem cells. We thought this may have potential through CD34 or CD45 stem cells which are enriched in this population, if they can rescue the amyloid beta burden artificially injected stereotactically into these mouse brains. So we sought to create this model and then followed it by enrichment of these stem cells derived from the cord blood using MAX and FAX, which is familiar to most of us as stem cell investigators, and try and study if these cells can survive and they can home on, and if they can differentiate, and if not differentiate, what kind of cytokine or growth factors they produce as a result of delivery of these kind of stem cells. So we are talking here of human cord blood derived lineage negative stem cells into the mouse brain. So there is a cross species thing which is also known to us if we review our literature. So we had these groups of mouse which were the group of mouse which were having amyloid beta deposits, the ones which wouldn't have amyloid beta deposits, and the sham controls, which were to see a 21-day of amyloid beta toxic results to come up. And on the 21st day we did the memory studies, and we put the stem cells of 2 doses, 50,000 stem cells and 100,000 stem cells, and then followed it up at 10-day and 60-day for Morris-Water maze, which is one of the traditional ways of looking at memory rescue as well as memory deficits. So that was the basic design, and we then tried to use these standard molecular tools, which is immunohistochemistry, real-time PCR analysis, and of course the Morris-Water maze, which I just mentioned, to look at what these stem cells do when they are transplanted and when they are not, which is basically PBS that we inject as a control. And that's the design. So we have day 1, which is the injection, and the day 21, which I just mentioned, is the transplantation of the cells, and then the 10-day and the 60-day analysis period. And of course, as is the recommendation by any ethics committee, and so is we have a very, very stringent ethical committee which mandates that we screen these cells for all the infections as it happens in all the cord banks. So they are stringently compliant to that. And we used a technique called the MAX, the magnetic stem cell sorting. We deplete out the lineage negative stem cells, so the lineage positive fraction is left out in the column, and the lineage. So we are able to get not only the lineage negative stem cell but also the lineage positive stem cells, in addition to the whole bone marrow soup coming out from the femur and the female, from the way the previous speaker described as the bone marrow cells are extracted from the bone marrow, from the bone. So this is just to show we used a dye and making sure that the stereotactic injection works well. And as you can see, this dye stains for the area where we delivered it. And then we tried to look at and also characterize the pathology. And the bottom right, you can see the FAS-L positive stem cells, FAS-L positive immunostain, showing that there is a FAS-L overexpression as a result of amyloid beta, all analyzed at 21 days after the amyloid beta was deposited. And you can see on the bottom, on the top left CA2 region that I just showed in the beginning, and describing it by zooming in into the brain sections. You can see the needle prick, which is used as a stereotactic means to deliver the amyloid beta. So the various stains, basically FAS-L on the bottom right, and C showing the Congo red, which actually stains for the amyloid beta deposition, as a marker that the model was in place and that the injury was indeed histopathologically being scored for. And then we've set our eyes on the lineage negative stem cells, which we, having isolated from the MAX column that I showed you, ran them through a FACS analysis, and those are the characteristic features of the cells that we used. And more interestingly, you might like to look at this data when we compared the expression of CD34 positive stem cells between the bone marrow derived stem cells on the left, and the middle, if you look at lineage positive stem cells and the lineage negative, you can see a relatively higher expression of CD34 stem cells in the human bone cord blood derived stem cells. So it's a CD34 expression far higher as compared to the MNCs and the lineage positive stem cells. And when you look at the bottom panel, you can see on the bottom right, the CD34 expression is also relatively higher as compared when it is compared between the lineage positive and the MNCs. But this is the bottom right one is the population of CD34 from the CD45 sorted cells. So it is also higher, as just like when you compare the whole bone marrow, not the bone marrow, the cord blood soup. The same trend seems to go with the CD117 expression. So the relatively higher lineage negative stem cell shows the higher expression of CD117 as compared to the MNCs and lineage positive stem cells, and which is also true for the CD45. If the same cells are looked for out of the CD45 cells which have been gated out. So, and this is the crux of the data, the main thesis of the presentation, of how you can appreciate that when there is a memory loss caused due to amyloid beta, I can see there's a consistent memory loss. You can see this here. I don't know if you're able to appreciate this. And when you have a cell dose of 50,000 cells at 60 days, so you can see a bit of rescue from the one when the amyloid beta is injected. And if you compare the 100,000, so these are the two cell doses. If you compare the 100,000 cell dose, 100,000 cell when analyzed at 10 day is able to rescue the memory loss. So what turns out from this slide is when you look at the behavioral parameters, the 100,000 cells, stem cells, at 10 days show as good rescue of the memory loss as the 50,000 at 60 day. So neither does the 100,000 cell dose work well at the 60 day, nor does the 50,000 cell dose work at the 10 day period. So that's the thing that we are looking for an explanation, and I'll be happy to discuss that if you have any suggestions in the form of questions or criticism. And when we looked at the retrieval time, the similar trend was true for the same two doses that I just showed you. And that's a search error which correlates with the same data. And we also do, as any other water-maze experiment, you not only look at the escape latency time, which I showed in those graphs, but also look at the tracking the mouse uses, the path the mouse uses. So in order to discriminate between the swimming ability or the motor ability between mice to mice or group to group, and that also follows the same pattern. And we also looked at the labeled cells, because these stem cells were labeled with CFDA. So in order to make a case that yes, the rescue of memory loss was happening through the stem cells, we were able to indeed find that these stem cells are indeed localized to the area where they were injected. So they could survive up to 60 days. So 2 months after the cells were transplanted, you could see under the green filter, the CFDA cells were indeed found labeled. So those are additional markers just to make a case as to the pathology was being set in using amyloid beta, and that was being rescued. So foraged B staining is known to basically signal or score for the neurodegenerative changes that are being brought about by the amyloid beta, were found in CA3 region. And I'll quickly move on to show you how the amyloid beta deposit was scored in the cryosections. So this is the amyloid beta plus 50,000 at 10 day, 50,000 at 60 day. This is 100,000 at 10 day and 100,000 at 60 day, as compared to the amyloid beta control at 10 day and amyloid beta control at 60 day. So the cells were homing on, and they were being there, and they could be found surviving up to 60-day period. So we looked at different differentiation markers, namely nestin and GFAP, and unfortunately, we couldn't find any of those markers being upregulated or downregulated in the amyloid beta injected or the 50,000 or 100,000 cells transplanted, either at 10-day or at a 60-day period. So there was apparently no differentiation at 10-day or 60-day. So what did we actually find, and how was the memory rescue taking place? So we looked at least two among the other panel of cytokines and growth factors. We found at least two major growth factors. One is the CREB, which is a transcription factor, upregulated at 100,000, 50,000 at 60-day period, 100,000 at 10-day period. So this is for the 50,000 at 10-day period. This was upregulated so this was a biochemical estimation for CREB. And then when we looked at BDNF expression biochemical level, there was no change in any of the groups. When we looked at the BDNF expression at the real-time analysis, so this was mRNA extracted from whole brains, not just hippocampal brains, so the entire brain of the amyloid beta and the 50,000 10-day, the 60-day, 100,000 10-day and 100,000 60-day period compared. So we could find that the 50,000 60-day period showed far more mRNA expression for the BDNF as compared to the other groups. So as of now, I can say that the CREB expression and the BDNF expression is apparently highly upregulated as compared to the other groups. And this is a summary. I pretty much summarized what I said in different data sets that I showed you, that the 50,000 cell dose at 60 days shows, along with 100,000 at 10 day, an enhanced behavioral improvement after the amyloid beta induced memory loss is caused, and there is a CREB and BDNF upregulation in the 50,000 cell group, and nothing much to be seen or to be noted in the other data sets. So the work is ongoing, and we're still looking forward to using siRNA and inhibitors to BDNF and CREB and see if we can reverse the rescues which have taken place using the amyloid beta stem cell, lineage negative stem cell derived from cord blood. So with that, I bring an end to the presentation. I'll be happy to take any questions. The more important thing is that the entire set of experiments was done in a Quality Council of India recognized laboratory, which is the only laboratory in the country which is recognized by Quality Council of India, which means all the protocols are driven through a quality assurance unit in the lab, and all the experiments were blinded to the users, whoever was the experimenter. So those are the sheets just to show as to how we blind our samples and how we run our SOPs. And this is the lab that I come from. These are the students, and I do not have that particular student who's his name is Avijit, and that's the data that I presented. Most of the data comes from his PhD thesis. So I'll be happy to take any questions.
One question.
I was wondering how they measure that memory acquisition.
Memory?
Yeah, how do you measure that memory acquisition?
So it is traditionally used by what is called the Morris water maze. It is a circular tank filled with water, and it is artificially divided into 4 quadrants by using a thread. And there the pictures in the room where the tank is kept has a lot of pictures. So the mouse, while trying to reach a submerged platform, so we calculate the time it uses from the time it is dipped into the water to the submerged platform using the spatial cues. So every successive trial, the amount of time it takes to use the platform reduces. It's called the escape latency time, which I showed in the y-axis. So with every successive trial, the mouse will take less time, the control mice. But if it is amnesic mice, as was the case in our case, we use amyloid beta to be deposited in the CA1 region in the mouse brain. So it was not able to reduce its time using the spatial cues. So its memory was lost. When you use the stem cells, it was able to recover its memory loss, analyzed by the time it takes to reach the submerged platform. And not only that, also the effort it takes to the track it reuses, not only the time, but the track it uses too. So we videograph it, and that I showed in the contours and the kind of lines that you showed in that data. If I can go back again very quickly. So it's called Morris water maze. That's a way. And there are, of course, this is the way. So the mouse, whatever pattern it uses, whatever path it uses, you can actually map it. And by that you can tell if it correlates with the time it takes. So
just one quick question. So you're using a xenogenic model, right? I guess the question is that, do you know whether, I know brain is an immune privileged site, but the question if you use maybe match or somewhat not that extent of antigenic, disparity, whether you would get better results.
So I would use a question to myself answer the query I raised as to why 50,000 cell dose works better at 60 day and not at 10 day, and 100,000 dose works better at 10 days and not at 60 days. So probably what and we're going to test it. We've hypothesized that because these are derived from the human origin and then they're transplanted into, so if 100,000 cells are transplanted into a brain, probably they invite a lot of immunomodulatory reaction, and that is why it works. It doesn't work well enough at the 60-day latter time period, but it does well at the 10-day because shorter time frame probably doesn't induce much of rejection or immunomodulatory response. As for the 50,000, it works well. It doesn't work well at 10-day, but slowly probably is able to accommodate or titrate with the adjustments of xenogenic thing and able to invoke some sort of a rescue effect at 60 latter time point. But I mean, it's one way of approximating. I mean, instead of using a mouse cell, you use in a mouse model. It's better to use a human cell source into a mouse rather than using it in a mouse. You cannot use it in a human, so that's one of the
Certain. Okay.
So it is immune privilege. Yes, that's
Thank
also there. Thank you.
Congratulations.
Thank you.
Here is your
Thanks.
certificate. Okay.
Thanks.
So the next speaker.